Hello, illustrious and fantastic micro lecturer folks. I'm going to give you some basics about microbes and genetics. So, right here, I have a little intro passing of genetic material from one bacteria to another. That really works with any organism, unless it's asexual organism. Okay, and there's lots of those out there. So, when we talk about passing genetic information from one organism to another, it can happen in terms of transformation, transduction, translation, etc. And we're going to talk about a few of those in this presentation, and why they do it, and how it happens between bacteria and nature, and then also how we might do it in lab and why we do it. Um, and definitely, we are not going to get in great depth because each one of these examples, there's multiple methods for each one of these that I'm going to talk about. So, your textbook is probably going to have a much greater depth in terms of information. So, why do organisms exchange that genetic material? Well, primarily to increase diversity. Increasing your genetic diversity means your chances of survival are greater because you have an increased ability, most likely, to adapt to your environment and survive and procreate. So specifically here in this presentation, I'm going to just highlight transformation, transduction, and conjugation in terms of how genetic material travels from one bacteria to another. So here's a little definition. Again, you need to stop these presentations. You need to turn down the audio. You can. So when you think about change in a bacteria, because DNA has been transferred, especially when you hear that term in this slide, you'll see naked DNA. It's generated when you've just got fragments of that DNA, or maybe even plasmids, and they're released from most likely damaged or dying cells. And the DNA fragments are then taken up by a living recipient cell, and then they're recombined with the recipient cell genome. So it's kind of like you're taking a little batch of ingredients, mixing it with a new batch of ingredients to create something better. Okay, and that's one of the whys. You're contributing to that genetic diversity, you're increasing that diversity. In essence, you're attempting to make it better, whether it be in nature or in the lab. And some of the reasons we do transduction, transformation, and conjugation, I highlight in some of these slides. If you remember from general biology, you probably studied chromosomes, chromatids, the genes located on those. So highlighting those, finding the origin of those, maybe using one to insert to another, not necessarily species to species, but within species. If you've got a defunct piece of DNA and you're replacing it with something that works. So right here I have an old slide from an old PowerPoint. Back in the day when instructors, remember when we stood in front of you guys and lectured? I don't know if it's better or worse now, but so this is just highlighting transduction, conjugation, and transformation. And it's kind of, they're crude drawings, but I think they get the point across because it just shows how information, that genetic information, is going from one cell to the other. All right, so transduction specifically, this is where you have the transfer of portions of DNA from one bacteria to another, but it's through a bacteriophage. So you see that little guy standing on top of that blue cell, that virus. When you have a virus that infects a bacteria, we call those bacteriophages. Okay, so when you see, what's that little thing? It's not a virus. It's what? It's a bacteriophage. Okay, it infects bacteria. This is from an old PowerPoint I have, and it just gives a nice little, the cycle of transduction, how that transfer of DNA happens between one bacteria to another through those viral bacteriophages. Remember, you can download these presentations, you can slow them down, you can stop them so you can see all those images a little bit better. So just think, going from donor bacteria to recipient bacteria through transduction, which means there's an agent involved like a bacteriophage or like a virus, right? And just like before I mentioned, you're going to change the genetic characteristics of the recipient cells. They're going to take in that information. Hopefully it's going to increase their diversity and their survivability. And it's also similar to when you think about viral origin of some of the cancers. Like I know some people, all the commercials and stuff about HPV and how some of the cancers we have are linked to viral origins. So, when we talk about the transfer of genes from one organism to another, or from a host, or from the same species, um, we're talking in terms of, we're not just thinking, hey, here's cell division. We're thinking about how things are fused together to create something new. And I just give you some examples. What you can study genetic linkages that way, the origins of some viral cancers that way, etc. I'm going to move on to the next one, conjugation. So conjugation is when you have this exchange of genetic material between bacteria, and there has to be some kind of physical contact required. That's because that DNA is exchanged through this little thing we call pili. And in this little illustration I have here, like on the right-hand side of that slide, it shows this little thing coming out like a little needle. That is the pili, and that's the physical contact I was referring to, where you have that exchange of genetic material between the two. This is from an old PowerPoint that I have, a nice little slide. It just gives the process of that bacterial conjugation, how it's going from one bacteria to another through that pili. I often say that it's bacterial sex, and that always gets everybody's attention, right? And it's not really sexual, exchange information, but it's almost like this little mating process, because you have a donor cell and you have a recipient cell. By no means is it the same as meiosis and all that kind of stuff, but anyhow, the same reason that conjugation happens, it increases that genetic variation. Are we seeing some patterns here? Yeah, of course, right? Also, when we think about the other methods of bacterial exchange or genetic exchange, it kind of might represent this little evolutionary step when we talk about conjugation. Okay, so genetic engineering. What exactly is it? Why do we do it, etc.? Why do we mess with this uptake of naked DNA? And we take cells that are broken down and release that DNA, and then the cell-free DNA, we collect it and isolate it. Why do we do that stuff? And there's various reasons we do it, and it's really changed. Definitely when I think of as a kid, and we had not mapped the human genome. There was just a lot we hadn't done. So when you talk about clinical lab skills, microbiology, genetics, these are fields that change. Biostatistics, bioengineering, amazing stuff. So I'm going to highlight a few: fusion, amplification, recombination, what those are, and it's just little things that we might talk about a little bit in lab. So when we talk about genetic fusion, I think probably that makes sense, because usually we talk about cells going through fission. Here we're talking about fusion, where we have a combining. So you've got a location on a chromosome to another location on a chromosome. So you're fusing that genetic material. There's also protoplast fusion, where you have the same DNA genetic material and it's combined. So you remove the cell walls, release that DNA of two different types of cells, and allow them to fuse on their own. This is from an old PowerPoint I have from a class that I've taught in the past, and it just shows that little process, but it's showing it with plant cells. So it's not just bacteria that we do this with, just do it with more organisms. Gene amplification, it's when that material, obviously from the terminology, it's amplified. You use those bacteriophages and you induce reproduction to occur more rapidly in a host cell. And so this little image I have back here is just kind of a typical process of how that gene amplification occurs. So again, if you need to slow these down, etc., and stop them and look at them, I'm not going to go through this whole slide, but I got this from some kind of workshop or something, but I thought it was kind of cool because it's, the workshop was sponsored by different companies, and they had a list of why we do that. Another one I wanted to mention was recombinant DNA, where you've got the recombination, you're recombining them, DNA's formed from two different organizations. You're clipping a gene with enzymes, and you're putting another gene. So it's kind of like you're replacing you have something that's not working and you're replacing it with something that is working. So these are just some examples of genetics and microbiology. I hope it was useful and supplemental to your textbook information. Thanks.